Morning. Good afternoon. I have to say that Dr. Rutberg and I are really pleased to have an opportunity through the internet to share the work we've done with PZP-22 and to try and make it an understandable program that we've invested ourselves in. Dr. R, do you wish to say anything at this point?
Yes, thank you to Stephanie and Monique and the Botstiber Foundation for setting this up. And we really look forward to talking to everybody today. And thank you all for attending. So now
let's go.
Let's go. Okay. We first want to acknowledge some of the people and institutions that have supported us since we've been doing this for, in John's case, something like 40 years, mine a little bit less than that. This is only a small sample of the population of agencies and people who helped us out. So we want to acknowledge those efforts. For today's webinar, we want to particularly acknowledge Taylor Grams, Kim Frank, and the late Dr. Baker Patrick from the Science and Conservation Center, as well as Erwin Liu, Doug Flanagan, Rick Nogel, and Kaylee Prera, who've been involved in the deer and the horse work over the years. So, this is how we come at things. We support the mission of the law and the Bureau of Land Management in the attainment of healthy rangelands with a thriving ecological balance. Somebody's really noisy. I don't know who that is. So everybody can hear themselves. But our particular interest as well is in the horses and wild horses living out a normal life as much as possible on the range, which means we are pursuing minimizing removals, and we are interested in developing and testing contraceptives that really minimize the impact of the treatments on the behavior and the wild lives of the horses. So the key messages today are not surprising, that we do believe that fertility control is essential for managing wild horse populations and maintaining the land. Our experience and the experience of the agencies is that removals alone are not enough to do either of those things, either to control the population or to maintain the state of the land. And in particular, having watched this agency and these horses over 30 years, the BLM has achieved or come close to achieving what it considers its appropriate management levels on a number of occasions. But every time they get close, something happens. There's either fire or drought, there's resources to gatherers. There's a change in administration or a turnover in personnel. Institutional memory is lost, continuity is broken. There are lawsuits, there are changes in congressional appropriations, and the cycles of gatherers are broken. And with a reproductive rate, with a population growth rate of anywhere between 15 and 25% for some of these herds, any sort of a break in the population control quickly causes the population to rise rapidly, and it becomes very difficult for the agency to recover in terms of its management. And what I see as an important role for contraceptives, whether it be PZP-22 or others, is to provide resilience to the system, to understanding that gathers do not necessarily occur at regular intervals, to keep the herds from growing so fast that the agency can no longer recover and the land can no longer recover.
So, Alan, I think I'll start, okay?
Your turn.
Good. It's fun taking turns. When I was a kid, I was always told in school, it's very important to share things with others. So the first thing we'd like to do is spend a little bit of time going back. There's many people probably on this webinar who know the background of PZP, but I'm going to give a little background and a little bit of the historical aspects of it and some of the early studies that we did. And then I'm going to turn it over to Dr. Rutberg again. So porcine zona pellucida, that's what PZP stands for. In the upper right of this screen, you see this circle, which is a micro photograph of an ovulated mammalian egg, and the little streamers coming off it are sperm that have attached to the egg. And so this egg has around its surface a protein shell, and that is our focus. What we've done is we extract the proteins in that surface from pig ovaries, and then we prepare the vaccine with a boosting adjuvant, and the boosting adjuvant then causes the female recipient of a different species to produce antibodies that block fertilization. It doesn't affect existing pregnancies, and there's no toxicity for horses or people. Alan. So how did we get started on this? Well, we had done some work for a number of years in the western US with horses in the Pryor Mountains in Montana and also in Idaho. But we became aware of the possibility of using a vaccine in the mid-1980s. And at that time, the National Park Service, not the BLM, but the National Park Service had become concerned about the fact that there were a growing horse herd on Assateague Island off the coast of Maryland that they were responsible for, with fertility rates ranging from 50 to 62% annually. And they asked if we would be interested in looking at this to apply a fertility control vaccine. We agreed. Park Service said you can't touch the animals, no handling. So this was all done with darting. We started in 1988, and it required 2 to 3 initial injections and annual boosters thereafter. But it was very successful. Fertility rate the first year was 0% out of 26 mares. Second year was 7%. And then by the time we reached 1994, the Park Service said it's time. We'd really like to take this over and make it our management tool. So between 94 and 2006, this slide shows you there was an average of 3.5% fertility. And if you look at the beginning in the 70s and early 80s, there was a fairly steep population increase curve. When we applied our first treatment, it slowed somewhat. And then in 94, the Park Service took over, and within 8 years, it had zeroed the population growth. And then thereafter, it began to decline. Now, this was done with an aggressive approach. In other words, all the mares that could be treated and accessed were treated. Next. So while we were in the middle of doing this work with Assateague Island, the National Park Service also had become interested in some early work we had done with captive white-tailed deer in Ohio. And we found in those studies that native PZP vaccine was very effective in white-tailed deer. So they invited us to come to Fire Island National Seashore on Long Island, New York, And between 93 and 98, we, using DART delivery, no capture or tagging, we treated the deer, as many as we could, on Fire Island. And we used volunteers for identifying individual deer. That was very successful because it turns out that the residents really loved doing it, and they became very involved with their deer. What we found using the PZP with annual boostering gave us a fertility rate of 5 to 15% in the treated does. And as you can see from the graph, it went from about 85% annually down to less than 10% on average. Next. So that was fine. Doing that work was excellent. We had a vaccine that would work at a high efficiency. We also knew that if we were going to go back to the Western states where we had started our adventure, where Jay Kirkpatrick and I had begun when we were looking at horses in the Western US as a possible target for controlling fertility in wildlife. Well, as you can see from those images, the areas there are huge. It's not like an island population. Secondly, the herd management areas in the Western states, in the 10 Western states, really have very few horses we would call approachable. Now, they can be water trapped and bait trapped, so they can be darted in smaller herds. But in the very large herds, which is a majority of the horses, it's difficult to get access. The BLM had been already doing a fairly long period of time in which they were using helicopters and doing their gathers with helicopters. And we recognize that this is stressful for both the horses and the people that have to do this work, but it was a routine procedure done about every 4 years, and we felt it would be a good way for us to have access to these to do studies. And our goal was that in order to do this, we would have to develop a one-shot vaccine that would have a multi-year effectiveness, because we couldn't be accessing these animals the way we were doing with the horses on acetate. So the first BLM researcher and Humane Society supported project in the field began in 1992 in the Dolly Varden Mountains in northeastern Nevada. And we had two groups. One group was given 2 shots of the native PZP vaccine a month apart, and the other group got a 1-shot native PZP vaccine with a controlled release component that would mimic what the boosters were. And we used initially very tiny BB-like microspheres, but we had some issues with them and they were not going to be a long-term solution. By the way, this is, can you go back, Alan? This is Dr. Liu. This is Jay Kirkpatrick, and he's also delivering this vaccine, and this is me. Okay, onward. So between 1993 and 2000, we studied various versions of this controlled release vaccine in a number of different field studies. We ultimately came up with and noticed that we had fairly effective, pretty good efficacy. This is an untreated control here, but we had fairly good efficacy over most of these herds. And the fertility rates after we were done went back to these levels. So one thing important that we had determined in both the acetate studies and in these field studies in the West was that this vaccine had a very important quality. It was reversible. When you stopped it after a while, the horses return to their normal fertile state. This implied a lot of potential for a long-term management tool because it gave flexibility. Next. We ended up with, after doing captive studies at UC Davis in 1998 and 99, with mares we had given this controlled release vaccine to, which eventually became PZP-22. And the reason it became PZP-22 was that in those UC Davis captive mare studies, the antibody titers that provided contraception were determined to be elevated sufficiently for a period of up to 22 months. So there we had PZP-22. What did it consist of? It was the native PZP. Go back, would you? One more. Thanks, Alan. So it consisted of the native PZP emulsion plus PZP and a different adjuvant that was water soluble, so it could be put into these pellets, which I'll explain in a moment. And the pellets were engineered to release at about 1, 3, or 12 months. And this shows you a dart with a probe that is pushing, that can push pellets into the needle barrel. This is a dime on this side here, and those little white dots are pellets. Those are the pellets. They fit inside a 14-gauge needle. Next. So the vaccine then has 4 components. PZP-22 has 4 components. And the pellet portion of the vaccine, PZP vaccine 22 vaccine has 2 primary components, which is the primer and the controlled release component. The pellets have 4 components. The 2 key ingredients are the PZP in a freeze-dried form, so it's a powder. QA21 is the name of the adjuvant, which is a well-established adjuvant, and it is also a powder. And what we do is we matrix these powders, we mix them together, and then we matrix them in a powders of 3 different types of polymers. Each one is designed to release over a different period. And then we heat them, the polymer with the vaccine in it, and compress it. And we also add a small amount of colored dye, a different color for each of the release characteristic pellets. So one of the important things about the pelleted portion of the vaccine is the ingredients are isolated, embedded in these polymers, and so they're stable. They survive to the boiling point and actually above it. They also have a long shelf life of about 5 years because they're prevented from being exposed to moisture, because moisture is what gradually degrades the polymer. It makes the polymer gradually break up into its components. So these characteristics made the pellet a very good approach as far as we were concerned. Next. We delivered the vaccine in field corrals initially, but we also had the capacity now to deliver it remotely in a dart. And this is what a PZP-22 dart looks like. You have the emulsion portion of the native PZP in the body of the dart. The barrel contains 3 different pellets. And when the dart impacts the hide of the animal, this causes the powder charge in the back to explode and push the plunger forward, expelling both the darts and the primer. Next. So with what we had in our hands, with PZP-22, that work that was done at Davis in captive animals, we went into the field with free roaming mares, and that was in the Clan Alpine Herd Management Area in central Nevada from 2000 to 2004. And we started with 100 mares that were treated. And of the 100 treated mares, we were able to track 67 of them across the entire 4-year period and keep track of them. So first year before treatment, 54% fertility rate. After treatment, 5. Second year, 15. Third year, double that. And by the 4th year, it was back to normal. Again, reversibility, very important. Next. So what I'd like to do now is say that we have this vaccine at a point where at this time, this was in 2004-5, where we were interested in learning more about it in the field, in the animals. And so with the Humane Society of the United States and the Annenberg Foundation together, a project was put together, an extended project from 2008 to 2016, actually. And the goals of this project were to examine the impact of PZP-22 vaccine on population growth, health, and behavioral effects. And so at this point, I'd like to return this to Alan, and he can tell you something about how this Annenberg project was run and what the results were. Alan?
Okay, thank you, John. With Kayla Grams doing most of the field work at Cedar Mountains and directing much of the others, we worked at 3 different sites. Sandwash Basin Herd Management Area in Colorado, Cedar Mountain Herd Management Area in Utah, and Harita Mesa Wild Horse Territory, which is a Forest Service territory in New Mexico. Mostly I will talk about the Sandwash and the Cedar Mountain studies. Okay, so one of the reasons that we were interested in doing this study is that we wanted to look at herd management areas that were a little bit more typical, since, as John mentioned, we were concerned with showing that we could actually have an impact on populations in more typical, more difficult herd management areas in the West. And so Cedar Mountain, just about 80 kilometers west of Salt Lake City, is a large and very challenging herd management area. About a quarter of it is wilderness, middle, which posed challenges of its own. Horse movements depended very much on the availability of water. And characteristically, the horses there were very wary. And at Sandwash is a little bit more tractable, as you can tell by the picture. Many of the horses are a little bit more approachable. And so, but it was also not an unusual kind of herd. So in Cedar Mountain, horses were gathered in December of 2008 and again in February of 2012. And they were treated by hand with PZP-22 in both of those gatherings. Sandwash Basin also gathered in October 2009, and that was the only gather. So after that initial gather, all horses that were retreated were boosted in 2010, were boosted by DART. So just to sort of get a couple things that emerged from the animal study in the way of efficacy. Harita Mesa, at Harita Mesa in New Mexico, Dan Elkins was able to deliver darts to corral-trapped, bait-trapped horses using the PZP-22 primers in in the darts. And it worked. And so demonstrating that dart delivery of PZP-22 is possible. We also delivered PZP-22 boosters in the Sandwash Basin, which also proved to be effective, with a little interesting asterisk. The primary efficacy of these studies was variable, not as consistent as at Clan Alpine, but at all sites it was effective for at least 1 to 2 years. The effectiveness was very closely related to timing. The efficacy was pretty high in the February 2012 gathers at Cedars, and the April to early June deliveries at Great Mesa, but less so at Sandwatch, the gather and the delivery of the of PZP-22 was in October, which is not really how the vaccine was designed. And there also were some issues with the pellets and the efficacy. One thing we found out that we were not expecting is that boosting these PZP-22 primed mares after 2 to 3 years yielded 4 at least additional years of contraceptive efficacy. And it didn't actually matter whether we boosted them with PZP-22 or native PZP. The native PZP boosters worked as well in terms of extending the period of. And so what that gave us is that we have at least 5 years of efficacy over about 7 years with 2 treatments of PZP-22 and a booster. And so averaging over Clan Alpine and the 2 Cedar Mountains gathers, which were consistent with the design time window, we saw roughly an 85% reduction in fertility in the first year, 63% reduction in fertility in the second year. It's pretty good. And I'll come back to that. A major point of our study, again, was looking at population. How did PZP-2 work in terms of affecting population dynamics in the field? So after the December 2009 gather, only about half of the mares on the range were treated. After the February 2012 gather, that was completed between boosting of the previously treated animals and the recruitment of new horses, PZP-22. Nearly 70% of the mares left on the range for the gather. It made a huge difference. What we showed overall is that 2 cycles of gathers really had a significant impact on the population growth. So here's the graph, white graphs. The blue line, which scales to the left, is the actual number of horses on Cedar Mountain. And so you can see that it climbs slowly during the course of the study, including the effect of a small gather in 2012, small that removed in 2012. The population growth over this study period was only about 11 or 12%, which is less than half of what would be a treated population. The gray line is the actual, which scales to the right side, is the annual growth rate. And so the best year we had was following the 2012 gatherings, where the annual population growth was a little bit more than 5%, which is very manageable. It rose slightly up. But clearly there was linked both to the effectiveness of the vaccine, but also to the proportion of mares. With 70% of the mares treated with an effective vaccine, we got some pretty good numbers. And so the lesson there is it probably will take more than 1 gather. It's not a one-off. But in a cycle of 2 gathers with persistent treatments and an serious attempt to gather most of the horses, you can have a huge impact on population. I'm going to divert to deer here for a minute because the efficacy data are really important to deer. As many of you know, among our participants today, the management of white-tailed deer, especially in the East Coast, but also surrounding a lot of Midwestern cities and suburban rings, is a pretty serious issue. The deer populations do very well in these areas and achieve pretty high densities and are associated with serious conflicts with human use, with deer vehicle and impacts on vegetation and so forth. And so we've been since Fire Island in trying PZP on deer. We've known since Fire Island that PZP, native PZP works well here, but we're also interested in trying a longer-acting vaccine for similar reasons as we use it for horses, which is to say, deer do not stand still every year in most places to get their shots. And so we have to assume that we're only going to be able to get it once or twice or maybe 3 times in its lifetime. And so that we started trying PZP-22 at Fripp Island starting in 2008. And at Fripp Island, we captured and treated deer by hand in February and March over a number of years. We've been duplicating that effort at Hastings-on-Hudson, New York, which is a very different one. And the success of the PZP-22 in both of those herds has been very similar enough. Like at Fire Island, in an untreated population of does, you get something like 80% of them having fawns every year. But in year 1, we got roughly an 85% reduction in fawning, so that we're only talking about 10% of those does fawning each year. And in year 2, about 20%, which represents 3/4 reduction. So we have definitely 2 years of efficacy with a hand-injected treatment that's really actually at a highly non-optimal time of year, since they're breeding in October, November, giving it to them. So it works really well in deer. In the past few years, Kaylee Prairie and I and an HSUS team have been working on looking at the effectiveness of boosters to see whether you see similar effects with deer as we do with horses. And our numbers are still pretty small. We're still doing that study, but it looks like we're going to see a pretty similar effect with at least 3 years of additional contraception with a single booster, either native or European. This is Fripp Island, and we were also, as we have been in horse sites, interested in population effects at Fripp Island. A resort community about 4 square miles. As it turned out, pretty isolated. We did not get much movement on or off the island. And there was an adjacent island that was a control site where there was no deer. That was Hunting Island, where there was no hunting, but that's what it's called. And we looked over 5 or 6 years at the fawn-doe ratios as well as the population density. And on the control site, the number of fawns per doe sort of vacillated over the years, but stayed at somewhere around 1.2 fawns per doe. Fripp Island, as we treated more and more does, got the number of fawns down significantly, so that there was only about 1 doe per 5, or 1 fawn per 5 doe towards the peak of the study. And at the same time, because it's a fairly isolated population, we got population densities declining very steadily at Fripp Island from a level about 2.5 times what it was at Hunting Island at the beginning to a level that was almost statistically independent. So just looking ahead a little bit in terms of managing suburban deer population, we know now that PZP-22 is super effective. So that we give it to them by hand initially and give them a booster 2.5 years later. Dart, we get at least 5 years of contraception, probably more. However, unlike horses, where mortality is very low, adult mortality of 2 to 3% at our Sandwash Basin study sites, you do see 10 to 15% of the female deer die or disappear in our suburban populations. You do not get, even in mainland populations, you do not get lots of new deer, lots of new females sloshing in. So while they're not strictly territorial, they're pretty site-based. Not get a lot of it. And so because of those 2 factors, we should be able to get substantial reductions of suburban deer populations within a 4 to 5 year treatment effort, with efforts of scaling over time. in a wide variety of environments, as long as the deer are accepted. Start them at least once, maybe twice, then you can make a difference. So now I'm going to back over.
Thanks, Alan. I'm going to finish up here by looking at, since our goal eventually has been, by whatever means, using something more than removals to help with managing wild horse populations, we specifically wanted to look at modeling into the long term. In other words, what would happen if you apply fertility control into the long term for managing wild horses? And we had found that with the Annenberg HSUS study, 2 treatments 2 to 3 years apart would give us a minimum of 5 years of effectiveness over a 7-year period. And we also knew that from the 2000-2004 data from the Clan Alpine study, that using updated PZP-22, that 2 treatments should yield 6 to 7 years of effectiveness. So somewhere between 5 and 7 years of effectiveness. Clearly way better than not doing. So what we did was we started looking at a database model of management of wild horses that would incorporate both removals and the application of PZP-22. And we chose a 50%, 50%. So when you do a removal, you can access in our models 80% of the horses on the range, of which half of those are removed, and the other half, the mares, are treated with PZP-22, and then the other half is released back onto the range. Next. So this is a particular model that we looked at as it approximates the number for an existing horse herd. And so we set it up with 680 horses, 80% captured, and we looked at what happens over a 10-year period using the database that we have for PZP-22 for an untreated population and a treated population. And when I say treated, again, it means that 50% of the horses are removed and 50% are treated with PZP-22 and released. And what you can see here is very obvious in terms of the number of horses on the y-axis, that when you just don't treat and then you just do removals, you get these large peaks and valleys. And a lot of that is due to the fact that the intervals between removals are long enough that new foals will mature and begin entering the reproductive pool. In addition, there is this effect called compensatory reproduction. When you remove animals from a population, the remaining animals will increase their reproductive rates. So if you're contracepting those, at least half of those animals that are back in that population, you are going to get fewer numbers of horses present after 10 years. And what did this look like when we took this model? Next. And we looked at the number of horses that would be removed at 10 years, over a 10-year period, and the cost, and we assumed an 80% capture rate for these things. Over 10 years, there would be 1,112 fewer horses from that 680 horse population that would be removed over 10 years. And when you look at the cost savings, it's huge because of the cost primarily of when you remove horses from the range, they have to go somewhere. It's not a lethal program. And so they are put in permanent sanctuary holding, which costs money. And based on dollar amounts published by the Bureau of Land Management, we created these graphs, which show a 10-year period with the use of 50-50 fertility control with PZP-22, saving $54 million over a 10-year period. Next. Because there are other vaccines that are being examined and studied, we wanted to look at the relative effectiveness over the time period of the 7-year time period that we initially had studied for an other vaccine. And the only one which has that length of time where there is published data reported for it, and we're looking at efficacy for treated mares from peer-reviewed published data. And Gonacon is the vaccine that has that. And we compared the two based on those published data over 2 years. So the published data from Gonacon and the data from for PZP-22 from Clan Alpine and the Annenberg HSUS project from Cedar Mountain, which is the closest resembling one to the Gonacon project, resulted in a difference between the numbers of horses. But the point is that whichever one you use, and assuming that Gonacon, which is continuing to be researched, will become an additional viable tool, is going to help to limit the growth of populations. So next. So our conclusions from the work with PZP-22 and related to other approaches to manage wild horse populations that involve vaccination. We know that research is continuing to make PZP-22, Gonacon, and other agents to be effective and simpler to use than they may be now. But the bottom line is that those other agents are still under investigation, but PZP-22 is completely vetted and it's reversible. In its current form, PZP-22 can be an effective management tool right now, while these other things are being explored and worked on. And so our view is that applying science, and that includes all of the efforts that are being undertaken by researchers, by the BLM, is what's best for the horses and the habitat. And that's what we started out wanting to do, what was best for the horses and the habitat. So all these years later, that's where we are. We still believe in the same principle. Use science as your guide. And because of that, our research group is extremely grateful to the agencies like the Bureau of Land Management, the Forest Service, the Annenberg Fund, the Humane Society of the United States, for contributing the funds and supporting this scientific investigation that's been going on for these years. We will have a solution that will make the lives of these animals what they should be and keep the habitat functioning properly. So thank you so much.
With that, we'd like to end the webinar. Thank you all for attending. And thank you, John and Alan, for
And on
participating
behalf,
and presenting today. Thank you so much.
you're welcome. In behalf of Alan and I, we appreciate that Botstiber has given us the opportunity to share.